What are the contamination risks for biology on the ISS? Have you ever wondered what happens to biology experiments in space? The International Space Station, or ISS, is a fascinating place for scientific research, but it comes with some unique challenges. One of the biggest concerns is contamination. Let's break down the risks associated with biology aboard the ISS. The closed environment of the ISS is a perfect breeding ground for microorganisms. Astronauts constantly shed skin cells and microbes during their daily routines. This includes activities like exercising, eating, and grooming. Despite strict cleaning protocols, these microbes accumulate on surfaces, in the air, and even in the water and food systems. Microbial contamination can include both harmless and potentially harmful species. For instance, some bacteria found on the ISS are known human pathogens. Examples include Staphylococcus aureus, Klebsiella pneumoniae, and Salmonella enterica. These microbes can become opportunistic pathogens, especially since astronauts' immune systems may weaken during long missions due to microgravity and radiation exposure. This means that infections can occur from microbes that would typically be harmless on Earth. Interestingly, microgravity affects how these microbes behave. Some bacteria may become more virulent or resistant to antibiotics after growing in space. This means that infections caused by these microbes could be more difficult to treat. The risks of contamination extend beyond health concerns. Microbial growth can degrade materials, contaminate water supplies, and interfere with scientific experiments. For example, biofilms, which are communities of microbes that stick to surfaces, can form on equipment and water systems. This can create maintenance challenges for astronauts. To manage these contamination risks, astronauts receive extensive training on hygiene and contamination control. The ISS undergoes regular cleaning and disinfection using chemical agents and methods like ultraviolet light. Research projects, such as the Microbial assessment of the International Space Station project, monitor microbial contamination on surfaces. This helps scientists develop better cleaning protocols and antimicrobial coatings. Spacecraft materials with antimicrobial properties are also being used to limit microbial growth. Sterilization techniques, including chemical sterilants and advanced methods like low-temperature plasma, are applied to spacecraft components before launch. This minimizes initial contamination and helps ensure the safety of astronauts and the integrity of scientific research. In summary, the contamination risks for biology on the ISS arise from human-associated microbes thriving in a closed, microgravity environment. These risks can lead to health issues for astronauts and operational challenges. Strict hygiene practices and ongoing research are essential to mitigate these risks and ensure successful missions in space.